Welcome to Podcast Learn and Fun. Let's start today's journey of learning. Today, we're going to talk about something really fundamental to life itself. How our cells manage to copy their DNA with incredible accuracy every time they divide.
Yeah.
Think about it. Every single cell in your body has a complete set of instructions, like a massive blueprint.
Right.
And somehow it manages to duplicate that entire blueprint with very few errors.
It's really quite mind-blowing. You see, DNA is this incredibly long molecule.
Right.
Shaped like a twisted ladder, the famous double helix.
Okay.
The rungs of this ladder are made up of pairs of chemical bases, the A, T, C, and Gs you might have heard of.
Yeah.
And the way they pair up is key. A always goes with T, and C always goes with G.
Right.
This complementary pairing is what makes accurate replication possible.
So when a cell decides it's time to divide, it needs to make two identical copies of this DNA blueprint, one for each of the new cells.
Exactly.
It's like making a perfect copy of a super detailed instruction manual before you give it to someone.
And to do this, the cell uses a whole team of specialized molecular machines. The process starts at specific locations on the DNA molecule called origins of replication.
Correct.
Imagine them as designated starting points for the copying process.
Right.
Then an enzyme called helicase comes in and literally unzips the DNA double helix, breaking the bonds between those base pairs.
Oh, wow.
This creates a fork in the DNA molecule, a kind of Y-shaped structure called the replication fork.
Okay, so we've got the DNA unzipped, ready to be copied. What happens next?
Well, that's where another enzyme called DNA polymerase comes in.
Okay.
This is the real star of the show.
All right.
It attaches to the separated DNA strands and starts adding new bases, matching them up according to those A, T, and CG rules.
Mm-hmm.
Essentially, it builds two new strands, each one complementary to one of the original strands.
So it's like DNA polymerase is reading the original DNA strand and then building a new strand by matching up the right building blocks.
Precisely. And it does this with remarkable speed and accuracy, but there's a catch. DNA polymerase can only work in one direction.
Okay.
So on one of the strands, called the leading strand, it can move smoothly along, copying the DNA in one continuous piece.
That sounds pretty straightforward. What about the other strand?
The other strand, called the lagging strand, is where things get a little trickier.
Okay.
Because DNA polymerase has to move in the opposite direction on this strand, it can't copy it continuously.
Right.
Instead, it has to work in short bursts, building small fragments of DNA called Okazaki fragments, which are later joined together.
So it's like building the lagging strand in a patchwork fashion.
Yeah.
It sounds pretty complex.
It is, but the cell has evolved this intricate system to ensure that both strands get copied correctly.
Wow.
Another enzyme called DNA ligase acts like a molecular glue, sealing up the gaps between those Okazaki fragments and creating a continuous DNA strand.
So we've got unzipping, copying, and gluing. But with all this molecular machinery at work, how does the cell ensure that everything is copied accurately? I mean, we're talking about billions of base pairs here.
That's a great question, and it gets to the heart of why DNA replication is so fascinating.
Yeah.
The answer lies in a multi-layered system of checks and balances, like an incredibly sophisticated quality control system.
Okay. I'm intrigued. Tell me more about this quality control.
Well, first off, DNA polymerase itself has an amazing ability to proofread its work as it goes along.
Oh.
If it accidentally adds the wrong base, it can actually backtrack, remove the incorrect base, and replace it with the right one.
Wow.
It's like having a built-in spell checker for DNA.
That's incredible. So DNA polymerase is like a master builder and a meticulous editor all in one.
You could say that,
Mhm.
but even with this impressive proofreading ability, sometimes errors can slip through.
Oh, no.
That's where a second layer of protection comes in called mismatch repair.
So it's like having a second set of eyes to double check the work.
Exactly. After DNA replication is complete, specialized proteins scan the newly synthesized DNA looking for any mismatched base pair or small errors that were missed during proofreading. And if they find a mistake, they can actually cut out the incorrect section of DNA and replace it with the correct sequence.
Wow.
It's like a molecular editor making corrections to a manuscript.
Wow, that's impressive. 2 levels of error correction. That's some serious dedication to accuracy. So with all these checks in place, are there ever any mistakes in DNA replication?
Well, despite this incredible precision, errors can still happen, though they're incredibly rare. And when they do happen, they're called mutations.
Mutations, those are the changes in the DNA sequence that we hear about.
That's right. Mutations are essentially alterations to the DNA blueprint. They can involve changes to a single base pair called point mutations, or even larger insertions or deletions of DNA segments.
I imagine these changes can have a big impact on how the DNA instructions are read.
Absolutely. Some mutations might have no noticeable effect, while others can lead to significant changes in protein function, potentially causing diseases or affecting an organism's traits.
So it's like a tiny typo in a massive instruction manual. Sometimes it's inconsequential, but sometimes it can throw the whole thing off.
That's a great analogy. And while we often think of mutations as negative, they can also be a source of variation that drives evolution.
That's a really interesting point. So mistakes in DNA replication, while rare, can have a profound impact on life itself, both for better and for worse.
That's the fascinating thing about DNA. It's a molecule that holds the secrets to life, and understanding how it replicates is key to understanding how life works and how it evolves.
It's amazing to think that all of this is happening inside each of our cells right now.
Right.
It makes you appreciate the complexity of life at the microscopic level.
Yeah.
But speaking of complexity, are there other factors that can affect the accuracy of DNA replication?
There are. Think of DNA replication as a finely tuned machine with lots of moving parts.
Oh, yeah?
The speed at which this machine operates can influence the accuracy of the process.
Oh,
If replication happens too quickly, there's a higher chance of errors slipping through.
that makes sense.
Yeah.
Rushing a job often leads to mistakes. So does the cell have a way to control the speed of DNA replication?
Absolutely. There are proteins that act like speed regulators, slowing down or speeding up the process as needed. It's like having a built-in cruise control system for DNA replication.
Wow, so it's like this intricate dance of molecules all carefully orchestrated to ensure that the DNA is copied correctly.
Exactly. And it doesn't stop there. The environment can also play a role in DNA replication.
Oh.
For example, exposure to certain chemicals or radiation can damage the DNA molecule.
That's a bit worrying. So even with all these safeguards, our DNA is still vulnerable to external factors.
It is, but it's also remarkably resilient. Cells have evolved an array of repair mechanisms to deal with this kind of damage.
Okay.
It's like having a team of tiny mechanics constantly working to fix any dents or scratches in the DNA.
It's incredible to think that our cells have these repair crews on standby at all times. But are there limits to what they can fix?
There are. Sometimes the damage is too severe or the repair mechanisms themselves are faulty.
Right.
This can lead to mutations that accumulate over time, potentially contributing to aging and diseases like cancer.
That's a sobering thought. It makes you realize how important these DNA repair mechanisms are for our health.
Absolutely. They play a vital role in maintaining the integrity of our genetic information throughout our lives.
Speaking of aging, we talked earlier about telomeres, those protective caps at the ends of our chromosomes.
Right.
I know they're somehow linked to aging, but I'm not exactly sure how.
That's a fascinating area of research. You see, every time a cell divides, its telomeres get a little bit shorter.
Okay.
It's like the tips of shoelaces fraying with each use.
Uh-huh.
Eventually, when the telomeres become too short, the cell can no longer divide properly, and it enters a state called senescence, where it stops dividing altogether.
Right.
This process is thought to be a major contributor to aging.
So telomeres act like a kind of biological clock, limiting the number of times a cell can divide.
Precisely. It's like a built-in countdown timer for cellular lifespan.
But I've also heard about an enzyme called telomerase that can actually add DNA back to telomeres.
Right.
How does that fit into the picture?
That's where things get really interesting. Telomerase is like a molecular fountain of youth for cells.
Oh,
It can replenish the telomeres, allowing cells to keep dividing beyond their normal limits.
that sounds amazing. Could telomerase be the key to slowing down aging?
It's a possibility that scientists are exploring. However, there's a catch.
Uh-oh. What that?
While telomerase activity is crucial during development, and in certain cell types like stem cells, in most adult cells, it's turned off.
Why would it be turned off if it can help cells live longer?
Well, it turns out that telomerase can be a double-edged sword.
Oh.
While it can extend cellular lifespan, uncontrolled telomerase activity is also a hallmark of cancer.
So it's like having a powerful tool that could be both beneficial and dangerous, depending on how it's used.
Exactly. If telomerase is reactivated in the wrong cells, it can allow them to divide indefinitely, leading to tumor formation.
Oh, no.
This is why researchers are trying to understand how telomerase is regulated and how it might be manipulated safely for therapeutic purposes.
It seems like there's still so much we don't know about telomeres and telomerase.
There is. It's a rapidly evolving field of research with the potential to revolutionize our understanding of aging and cancer.
This whole conversation has been incredibly eye-opening. It's amazing how much is happening at the molecular level to keep our cells functioning and our DNA intact.
It really is. DNA replication, with all its intricate mechanisms and safeguards, is a testament to the power of evolution and the incredible resilience of life itself.
It really makes you realize that we're not just walking, talking beings, but incredibly complex ecosystems of cells all working together in ways we rarely think about.
It's a humbling thought, isn't it? And the more we delve into the molecular world, the more we discover just how much we still don't know.
Speaking of unknowns, what are some of the big questions that scientists are still trying to answer about DNA replication?
One of the biggest mysteries is how the entire process is regulated so precisely. We know many of the key players, the enzymes and proteins involved, but the sheer complexity of the system is mind-boggling. How do all these different molecules coordinate their actions so seamlessly?
Yeah.
How do they know when to start, when to stop, and how to adjust their speed based on the cell's needs? These are questions that still keep researchers up at night.
It's like trying to figure out the intricate workings of a giant invisible machine with millions of tiny parts.
Exactly. And beyond the mechanics of replication, there's the whole question of how it relates to aging and age-related diseases. We know that telomeres shorten with each cell division, but what are the downstream consequences of this shortening?
Right.
How does it contribute to the decline in cellular function that we see as we age?
Yeah.
And are there ways to intervene in this process to slow down or even reverse the effects of telomere shortening?
I imagine that researchers are also exploring the potential of manipulating DNA replication for therapeutic purposes.
Absolutely. One area of intense focus is cancer research. Since cancer cells are characterized by uncontrolled cell division, targeting the replication machinery in these cells is a promising avenue for developing new treatments.
So the idea is to essentially disable the copying machine in cancer cells, preventing them from multiplying.
Precisely. Another area of exploration is gene editing, where researchers are developing techniques to make precise changes to the DNA sequence. Imagine being able to correct genetic defects or even enhance certain traits by directly editing the DNA blueprint.
That sounds like something straight out of science fiction.
It does, but these technologies are rapidly advancing. And at the heart of it all is a deep understanding of DNA replication, how it works, how it can go wrong, and how we might harness its power for the benefit of humanity.
Well, I think we've given our listeners a lot to ponder today. It's been a fascinating journey into the microscopic world of DNA replication, and it's clear that there's still so much more to learn.
It's a field that's constantly evolving with new discoveries being made all the time, and who knows what incredible breakthroughs await us in the future.
Thank you for listening to Podcast Learn and Fun. If you like our videos, please subscribe, click the like button, comment, and share. See you next time.